Welcome to Carnegie. I am Eric Isaacs, the president of the Carnegie Institution for Science. And thank you all for joining us tonight to hear the Kavli Prize Laureate lectureship tonight, which is going to be given by Carla Shatz, who won the prize in 2016 for her work in understanding how the brain forms connections. It's an honor tonight to do this, to co-host this evening with our colleagues, Dr. Eric Marshall, who is the Vice President for Prizes in Public Programs at the Kavli Foundation, and Dr. Øystein Hov, the Secretary General of the Norwegian Academy of Science and Letters. So to begin, I'd like you to join me in giving a warm welcome to Eric Marshall.
Good evening. It's wonderful to be here with you tonight, and I thank Dr. Eric Isaacs for co-hosting this evening's Kavli Prize Laureate lecture. Now, we've enjoyed partnering with the Carnegie Institution for Science for Kavli Prize Laureate evenings every year since its inception in 2008. Now, we're going to start by offering you a brief film to introduce you to the Kavli Prize, to the Kavli Prize partners, and the founder of the Kavli Foundation, Fred Kavli, whose vision led to the inauguration of these prizes. Now for the film.
What if we could understand the origin of the universe? What if we could improve lives by controlling atomic structures? What if we could truly grasp the complexities of the human brain? The story behind the Kavli Prize started in the 1930s with a curious boy named Fred, growing up among the tall mountains of Eresfjord in Norway. A deep curiosity about nature and the universe stayed with Fred throughout his studies in physics and his entrepreneurial success in the United States. Until he eventually established a philanthropic foundation with the vision of advancing science for the benefit of humanity. One of the first activities of this foundation led to the inauguration of the Kavli Prizes, beginning in 2008. The prizes are awarded every 2 years in a partnership between the Kavli Foundation, the Norwegian Academy of Science and Letters, and the Norwegian Ministry of Education and Research. Each of 3 international prize awards consists of $1 million and a gold medal received in a ceremony in Oslo, presided over by a member of the Norwegian royal family. The Kavli Prize laureates are selected by the Norwegian Academy of Science and Letters, based on recommendations from prize committees, comprised of leading international scientists from 6 of the world's most renowned science societies and academies, in the 3 scientific fields: astrophysics, nanoscience, and neuroscience. The biggest, the smallest, and the most complex. The Kavli Prize has 4 ultimate purposes: to recognize outstanding scientific research, honor highly creative scientists, promote public understanding of and appreciation for scientists and their work, and to foster international cooperation among scientists. Time and time again, we have seen that achieving these goals has been critical to making this world a better place. The Kavli Prize continues to be driven by the same sense of awe and curiosity felt by Fred Kavli as he grew up in the midst of nature at its most sublime, experiencing the vastness of the universe.
Thank you very much. And that was the U.S. debut for this short film. So the role of the Kavli Foundation has been to provide the award money and gold medals to the laureates and to help publicize the awards. The Kavli Prize is just one of many programs at the Kavli Foundation, including its basic support of basic research at 20 Kavli institutes located at major research universities around the world, and a series of Kavli symposium and meetings that facilitate critical dialogue among scientists as well as those that are connecting science and society. Now, 7 years ago in this very auditorium, Fred Kavli said, "We support science because practically everything that we touch in our daily lives has been improved or has been developed through basic research." So we're in for a great treat tonight as Dr. Carla Shatz, one of our 2016 Kavli Prize winners in neuroscience, and a Stanford University professor, will give a short talk and then engage in a conversation. First with Mr. Frank Sesno. Frank is director of the George Washington University School of Media and Public Affairs, and he is an Emmy Award-winning journalist. And then you, the audience, will then be invited to participate in this conversation. But first, I'm pleased to introduce Secretary General Øystein Hove of the Norwegian Academy of Science and Letters, and I thank you so very much for sharing this evening with us.
Thank you very much. As the Secretary General of the Norwegian Academy of Science and Letters, I'm happy to be here tonight, and it is an honor to introduce Carla Shatz as the lecturer. She was one of the three that shared the Kavli Neuroscience Prize in 2016. And here is the evidence for that. These are the three prize winners, and here they are, all of them, for the three prizes, together with the Crown Prince Haakon in the Royal Castle in Norway. Carla Shatz is one of a growing line of eminent Kavli Prize winners. The role of the Norwegian Academy of Science and Letters is to take care of the nominations and selection of the prize winners, to decide on the winners and organize the award ceremony. And the Norwegian Academy is happy and proud to be a Kavli Prize partner, together with the Norwegian Ministry for Education and Research and the Kavli Foundation. Through our work with the prize together with the partners, we use the Kavli Prizes in our efforts to promote the public understanding of science and the appreciation for scientists and their work. This is more than ever an important task in society and a core mission of an academy like ours. The work with the Kavli Prizes continues, and here we have this year's winners, with the King Harald V of Norway, taken 4 weeks ago in Oslo. And here they are, the names of this year's winners. And here we have a little commercial in the end. Thank you.
Thank you, Eric. Thank you, Øystein. Now it's my privilege to introduce to you a little bit about who Carla is, this extraordinary scientist. The Kavli Prize in Neuroscience recognizes the extraordinary work that is being done today to advance our knowledge of the brain and the nervous system, how they form, how they function. Over the past 40 years, our understanding of the brain's architecture has been transformed in no small part because of Professor Carla Shatz. Previously, most neuroscientists believed that our brains lose capacity to grow and form abundant new neural connections by the time we enter adulthood. Dr. Shatz and her colleagues have shown that the adult brain is actually much more flexible than had previously been believed. In our research, she's uncovered some of the mechanisms that strengthen or prune an infant's myriad neural connections, and in particular, focused on the region of the brain that receives visual information and the ways that our visual systems can alter both the architecture and the functioning of the brain. And it turns out that many of the mechanisms that Dr. Shatz discovered hold true for development of other pathways in the brain, not just the visual pathways. This work has, as you will hear tonight, great implications for our understanding of neurodegenerative disorders such as Alzheimer's disease. A little background, Dr. Shatz is currently at Stanford University, and she serves as the Sapp family Provostial professor and professor of biology and neurobiology. She also is the director of BioX, which is Stanford's interdisciplinary biosciences institute. She earned her undergraduate degree at Radcliffe College and was awarded a Marshall Scholarship to study at University College London, earning a master's in philosophy in physiology. She then went to Harvard, where she became the first woman to earn a PhD in the Department of Neurobiology. In 1978, Dr. Shatz joined Stanford University School of Medicine in the Department of Neurobiology, where she began her research on the development of mammalian visual systems. She became professor of neurobiology at Stanford in 1989. In 1992, she moved her laboratory to the Department of Molecular and Cell Biology at the University of California, Berkeley, where she became a Howard Hughes Medical Institute investigator. In 2000, she left the West Coast to lead the Department of Neurobiology at the Harvard School of Medical School, becoming the first woman ever to lead a basic science department at the medical school. While at Harvard, she helped to develop the Harvard Center for Neurodegeneration and Repair, which is now called the Neurodiscovery Center, and led the Harvard Center for Brain Imaging. She then returned to Stanford in 2007. A lot of traveling. Dr. Shatz is a member of the American Academy of Arts and Sciences, the National Academy of Sciences, the European Academy of Sciences and Arts, and the Royal Society in the UK. In addition to the Kavli Prize, her awards include the Gill Prize in Neuroscience, the Mika Salpeter Lifetime Achievement Award, the Ralph W. Gerard Prize in Neuroscience, the Gruber Prize in Neuroscience, and the Harvey Prize in Science and Technology. Please join me in giving a warm welcome to Dr. Carla Shatz.
Well, I'm delighted to be here. It was a lot of back and forth, it's true. That's a story for another day. But it's a great honor to be here today and really to celebrate science together with you. I resonate with the themes and the goals that we've just discussed with the Kavli Institute and the Carnegie Institution. And it's just great to be here. So thank you, Eric, and thank you, Austin, and thank you, Eric, for convening this wonderful event. So the question that I want to consider is, what can the developing brain teach us about Alzheimer's disease? But another question we could ask is, what is a developmental neuroscientist, somebody like me who studies brain wiring? What in the heck am I even doing asking this question of you? Well, I have to say that one reason is that I'm worried. And there are a few reasons for this. So this is a statistic, a fairly sobering statistic. And I think it's still very true today, even though it was collected in 2008. These numbers have not gone down, which is that the Alzheimer's disease frequency doubles every 5 years after the age of 60. And of course, there are a few of us are going to be very lucky, and we're going to have healthy aging. We're going to be one of those unique people who will live until we're 100 and have wonderful cognition up until then. But I worry about that, and I also worry because in fact, many of the drug trials that have been really assiduously pursued over the last 10 years or so have yielded very disappointing results so far. So I'm getting older, even though I'm a developmental neuroscientist. It occurred to me that maybe if we could understand molecules and mechanisms regulating developmental critical periods, and I'll tell you what they are in a minute, we could use that knowledge in age of brain repair and also perhaps to treat Alzheimer's disease. And I found myself in a rather surprising position to think that maybe some things that we understand about the developing brain really could be relevant. Now, what is a critical period? Well, you know, one really nice thing is if only old Mickey could become young Mickey again. And I always think, well, it'd be really great to have a pill that I could take. And if I had that pill, what I would do as an older person is I would finally learn how to speak French without an accent. Okay, this would be cognitive enhancement. This would be great. So that's a great example of a developmental critical period. Language learning. We all know how easy it is for the baby's brain and for, you know, young children to learn a language. In fact, they can learn one language, they can, you know, learn their native language, they can learn two, they can learn three, it's no problem at all. Whereas after about puberty, it becomes more and more difficult to learn languages. And in fact, wouldn't it be great if we could understand enough about that developing brain to be able to reopen that kind of developmental plasticity as adults, you know, for language, but also maybe for brain repair. So a critical period is a period during development when exposure, practice, and learning of a particular task is required in order to fully master that task. After the critical period is over, of course we can still learn, and I can still try to learn French, and I can limp along, no problem. So, but the nature of learning has changed afterwards. And so what is different about the baby's brain and my brain, for example, as an adult? That's really the question I'm very interested in answering in my lab. Now, how could you go about really studying molecules and mechanisms of developmental critical periods as a scientist? Well, last thing I heard, Mickey Mouse was not speaking French. So the question is, what kind of a system could we study in an animal model like a mouse, where there is a developmental critical period? And then we could learn about the mechanisms of that developmental critical period and maybe even find molecules. And it turns out that one such system, even in the mouse, is the visual system. So here's a question for you. Why don't we see double all the time? I mean, some people see double with pathology, but the rest of us are lucky enough to be able to have perfectly good, what we would call a cyclopean, a single view of the world, even though our eyes are sending input to the brain with two views of the world, one in the right eye and one in the left eye. Well, it turns out that the brain actually has to learn how to use both eyes together during a developmental critical period. And I want to just tell you a little bit about that now. So the way that the brain brings both eyes together so that we have one beautiful uniform view of the world is really in its wiring. And let me just give you a kind of simplified version of wiring in the visual system that's relevant for binocular vision. So, for example, the output neurons of the eyes, which are called the retinal ganglion cells, and there will be an exam on this later, but it'll be okay. RGC is a good answer too, so that's fine. So the output neurons from the eyes, they're taking visual information that's been brought to them from the rods and the cones in your eyes and sending it to the rest of the brain. And you can see that part of the answer for why we don't see double all the time already starts right here at this first sets of connections between the retinal ganglion cells and the first relay target structure in the brain, which we're also going to be examined on, called the LGN. Now, the next step that happens is the neurons in the LGN send their connections up to the primary visual cortex. And you can see immediately that there are these little gray nerve cells that get input both from a right eye red neuron and a green eye left neuron. And that is sort of the really the bringing together and the intermixing of the inputs from the two eyes to create binocular vision, so a unified view of the world. And it's very important to have this view, because without it, we actually don't have stereoscopic depth perception and other really important aspects of the vision that we as primates have. And that's a whole nother topic for a whole nother lecture. But I've simplified this drawing just to make a few points. The first is that there are about 1 million ganglion cells in each eye, and there are about 2 million target neurons in the brain. So this is very simplified, but the bottom line is that there's this beautiful segregation of input at the level of the LGN. So right eye ganglion cells send their connections to right eye LGN neurons, left eye ganglion cells send their connections to left eye neurons. And this sort of beautiful alignment of the input from the two eyes is the first step in wiring. Now, what do I mean by connections? Well, this is really an important point. And, it turns out that if you remember anything about my lecture tonight, your brain will have changed, because genes in your brain that are learning genes will have turned on, and they will have laid down memories. I'm going to get back to this in a few minutes. But the one thing I really want you to remember tonight is the synapse, the idea of a synapse. This is the junction or the connection between nerve cells, and it allows a nerve cell to relay information from one nerve cell to the other. And just to spend a minute talking about what the synapse is, information is collected by nerve cells by receiving sides of the nerve cells, which are called dendrites. And there's a lot of computation that goes on in the neuron. If you could say, why do you have to have synapses at all? The reason you have to have synapses is because there's steps where computation, where adding and subtraction and division and multiplication go on in the nervous system. So the nerve cell takes this computation and the result of the computation and sends it down its axon using very beautiful signaling, which is called the action potential, and it's a chemical electrical signal that's sent over long distances. And when that signal gets to the end of the nerve cell, then chemicals are released, and these are the famous neurotransmitters. So this is where the drugs of abuse happen, right here at the synapse, because they interfere with this chemical neural transmission, or they can actually augment that chemical neural transmission. The chemicals are released, and then they in turn influence the computation of the next neuron along the line. Now remember, there are going to be now billions and billions of neurons in the brain doing this kind of competition. And then computation. And then this computation is then passed on along the next nerve cell, the axon, to its synaptic terminals, and so on. Now, why is this so important? I mean, you can call them connections. I don't care what you call them, but the bottom line is that you are your synapses. And what I mean by that is that if our synapses were all the same, in the same pattern, the same nerve cells, we'd all be like computers and automatons. And by the way, we also would never be able to adapt or learn or anything. So our synapses change with learning, actually. They get stronger, or they even get weaker, forgetting. And your memories are stored at synapses in these circuits. So the synapse is an extremely important aspect of the nervous system, in fact, quite unique to the nervous system. So information is relayed from the eye to the visual cortex via these LGN neurons, via these synapses. And it turns out that these connections from the eye to the LGN form very, very early in development. In fact, so early, even in us in utero, even before we have our rods and cones and before we can see. So because of that, people thought that this beautiful eye-specific layer of green and red inputs had to form in a hardwired pattern with a very specific set of genetic instructions that would tell the nerve cell axons to connect from the beginning in the right pattern. By the way, amazingly, the eye is not even connected to the brain initially. And those connections have to form. And then they have to go to the right place, and then they have to form these layers. So in my lab, we thought, let's check this out and let's see if that's really true, that these beautiful eye-specific layers form from the beginning. And so what we did is we actually labeled and traced these very connections from the eye to the LGN in, for example, mice models. And we were extremely surprised to find that the adult pattern of connectivity isn't even present initially in development. And it only emerges through a period of development, which in all mammals, including us, happens before vision. And I'll get back to that in a minute because it's quite mysterious what's going on here. So the immature pattern of connections, so these synaptic connections, are such that a given target LGN neuron receives input early on from retinal ganglion cells in both eyes. Then there's a kind of remodeling that goes on. And finally, the adult pattern emerges in which the green eye layers get input from the green eye and the red eye layers get input from the red eye. Now, this remodeling process involves a mixture of the pruning and removal of some synapses and the stabilization and growth of other synapses. And the key word here is pruning. This is what I want to talk about in the context of Alzheimer's disease. So this transformation happens because this eye's connections are, these synapses are actually pruned from here, stabilized here. The red eye synapses are pruned from here, stabilized here and here. So this remodeling is actually what gives rise to the adult pattern of connectivity. This was a huge surprise. We can talk about how hard it was to get people to accept this finding at another time. But in any case, it was a huge surprise to us and to everybody. And here's another thing that was a big surprise. The surprise was that this pruning process and remodeling process actually requires signaling from the eye to the brain. And so I've sort of drawn that here with these yellow, wake up eyes. Okay, so this signaling didn't make sense either, because the signaling is just what I told you. It's the sending along these long axons of chemical electrical signaling from the neurons in the eye to the synapse in the LGN. But I already told you that that's ridiculous because that signaling is happening way before the rods and the cones are present. It's happening in utero. It's happening before vision is possible. So the other thing we discovered is the eye is actually spontaneously signaling to the brain, almost like running, if you were an engineer running a test pattern on a circuit you just built. That's what the eye does. These ganglion cells are what we call spontaneously active, sending signals to the brain very early in development, and those signals are used to test which connections should remain and which ones should be pruned. And we discovered that if we block this signaling process from happening, then these beautiful mature layer organization does not emerge. Instead, there's a persistence of this immature pattern. Okay, now I'd like to argue that this is a great example of an early developmental critical period, namely a period during which the connections from the eye to the brain critically require this signaling process in order for the connections to form properly in development. So in a way, this is a learning process. The eyes are actually learning how to get themselves organized in the brain during this developmental critical period. And this process of synapse pruning happens not just from eye to the LGN in development, but it happens throughout the brain during development, not only in utero, but also for extended periods of time in us during our postnatal lives. And a wonderful example of that is, again, just to look at the development of synapses in the cerebral cortex of the visual system. That's the next step in visual processing. And what you'll notice here is in the human brain that early on, there's a lot of growth of synapses. Then there's a period where there's actually a decline in the total number of synapses or their density. And that is due to the fact that pruning at this time is greater than growth. And you'll notice this is kind of an extended period of pruning, and then it kind of levels off. And in other parts of the brain, like the frontal lobe, this pruning process, this even goes for much longer. All right, so what does all this have to do with Alzheimer's disease? And that's what we really want to talk to you about tonight. And I think I'd like to invite Frank Sesno to pop up, and maybe he'll even ask us this question. And I'm going to leave this thought and also a complicated slide here, just in case we want to talk about what are some of the hallmarks of Alzheimer's disease as we move along. So, and this is time for you also to really think about your questions. And I think, Frank, why don't you come up and we can chat.
All right. Thank you.
Yeah. You're welcome.
Well, first, let me say what a privilege and pleasure it is to be here with you, Professor Carla, and with all of you here. What a great audience and what a great opportunity for us to have this conversation. I don't know if this is possible, but I've been trying to exercise my synapses in preparation for this because this is a little scary. But let's start with your question,
Sure.
Which is, what is the connection to Alzheimer's? You've got a tremendous trajectory, and you've been working on this really all your life. So what's the answer?
The answer. Well, you know, I think I gave you my answer. And I think it's really worth thinking about this in depth.
Sure.
I think partly the answer is pruning. So remember, I told you that you are your synapses and that memories are stored at synapses. And during these developmental critical periods, this kind of pruning process is going on normally. And why is it happening normally? It's happening normally because this is a kind of use it or lose it period in our brain development, when connections that are formed are stabilized with use. And you know, again, we can go to language. Language learning is a great example.
Right.
You know, if you hear and learn a few languages, that pruning process is selectively eliminated at synapses that are being used, so you can actually build circuits not only for your native language, but we think also for other languages that you learn. If you don't, the concept really is that the connections are waiting to be used, and if they're not fully used, they can be pruned away. So that's healthy, that's a normal, healthy development. But since learning and memories are stored at synapses, we can ask, what about Alzheimer's, where we know that memories are lost?
Right.
And there's, you know, major cognitive impact. And in fact, I think in many ways the most tragic aspect of Alzheimer's in late stages is that really you lose yourself because all of your memories, many of your memories are gone. So could Alzheimer's really involve dysregulated synapse pruning?
Dysregulated synapse pruning.
Yeah, so let's think about that. Because
what is dysregulated?
Yeah. So how come when we learn and remember throughout life, why don't our brains just explode through our skulls? Because
some might say we're threatening to have that happen these days, but that's another discussion.
So, okay, you know what I mean, right? If you keep forming and stabilizing as you learn, then, I mean, the brain grows, and you know the synapse part of all the neuropil grows, right? So forgetting is thought to be also an important part of learning, and part of that means that some synapses, particularly those that are not used all the time, may also be eliminated, i.e., they may be pruned. So throughout life, there's a combination, a balance between synapse stabilization and synapse pruning. So it's not like after these developmental critical periods are over, we stop pruning our synapses. In fact, they turn over slowly throughout life. So dysregulation could suggest that in disease, maybe too many synapses are pruned, and the balance between learning and adding and stabilizing synapses and the delicate pruning that otherwise happens in the adult brain is shifted, and there's excessive pruning, which would be perhaps a bummer, and that's what I mean by dysregulated, because I say that because really we don't know in the human brain all of these details. So that's what I mean by dysregulated.
You have looked at, and you've been captivated, we've talked about this, about how the brain wires itself during development, as you said. And you consider the child's brain going through this incredible sponge period where it's absorbing all this information, it's absorbing all this learning. And you've asked how and whether we can understand the molecular mechanisms that regulate the sponge periods. And this is the interesting thing, whether they can be turned on and off, right?
Yeah.
But what do you mean,
Yeah.
can we turn them on?
Yeah.
And how does that connect to the synapse
Pruning.
disconnect that's
Yeah.
happening in Alzheimer's or in other
Yeah.
cognitive challenged moments?
So, well, I guess a question that's always really fascinated me is, well, there are two questions. One, why, well, there are three, there could be four. You guys can ask, right? Okay, why do we have these developmental critical periods? Number one, we could ask that one. But if we have them, why do they end, you know? And by the way, don't just slam shut, you know, because as I said, we can still learn, but the quality and capacity of learning has changed by adulthood. So why do they end? And then can we reopen these developmental critical periods, kind of make the brain more juvenile and young-like for learning?
Now you're talking.
Right?
Yeah.
Okay, yeah, no. So like, okay, think about whether you want a pill or not, okay?
A pill.
Yeah, pill that we could take, right? And reopen this kind of developmental critical period. Now, what do you think, what would the pill do? How would you even find a pill? Well, you know the answer. I just told you the answer, because pruning is a process that definitely gets turned down. I mean, I showed you, I'm going to show it again. I showed you a graph, right? And here it is. So, there it is. Right? So here is a graph where this pruning is turned down and stabilizes. So what if we could figure out a way to block the pruning completely so that we could add back synapses so they could be available for French learning,
that's what the
example,
pill would do.
Right? And the idea would be if we knew enough about the pruning, we could somehow block it, and that would be a pill. Right?
Yes.
And then, so we did that in my lab, and we didn't teach mice to learn French, but but we actually we blocked the pruning and discovered that the mice could put new synapses back into their adult brains, and they could have their developmental critical period for vision restored. It was really amazing. And so then a light bulb went off for us, because we thought if we could do that, then maybe we could address problems of pruning in neurodegenerative disorders like Alzheimer's, where it's thought that there's excessive pruning and pruning is in overdrive. So that was the logic. And that's what brought us to think about Alzheimer's, as I said, not just to mention that I'm getting older and the drug trials are not promising.
So you developed actual experiments to block this signaling, correct?
Yes.
And you found that you can block it. Did you find then that you can turn it back on with these mice?
Yeah, so we can turn back on the critical period.
Right.
So we can make their older mice brains.
So what did you
more,
find that
yes.
those older mice were able to do?
They actually were able to do things that only young mice can do in the context of visual tasks. And one of the visual tasks that they could do again was to learn how to use both eyes together. So it's a complicated story, but what if you lose vision in one eye when you're young? Or if you have a weak eye, then the good eye is often patched, and so the weak eye can strengthen. And by the way, that is taking advantage of the plasticity, the early plasticity in the brain. So allowing the weak eye to get stronger again means it is actually, the connections are capturing synapses and blocking the pruning of those synapses. But if the weak eye is not addressed early, then, and the critical period for vision passes, and now you're an adult, it's not possible really to effectively restore vision in the weak eye. And often you lose binocular depth perception too. Sometimes the brain just turns the weak eye off.
Right. So you were able to restore that.
Yes, so we thought, let's test our approach in a mouse model of it's called amblyopia, but this is the weak eye story in us. So in mice, just by closing the eyelids briefly, they also get a weak eye. And if we then ask if that's because pruning has changed, we find the answer is yes. So then we thought, let's just see if, as an adult, we can restore vision through the weak eye by blocking pruning and allowing new synapses to appear that could be captured by using your weak eye, as if you're starting all over again in the critical period.
And if you can get new synapses to appear for your eyes, the presumption is, or the hypothesis is, that you can create new synapses to do other things?
Yeah, well, we could learn, like a pill, we could learn French.
You can learn your French.
Yeah, because maybe the idea here is that our synapses are very stable, and really, why shouldn't they be? You don't want to forget everything you've learned. So the amount of pruning that would make space for new synapses that would allow for new learning is very low.
Now, what people probably don't know about you as well, in addition to being an incredible neuroscientist, you're a bit of a rap star.
I am.
Cells that fire together wire together, and out of sync lose your link. These are your words.
Yeah.
You want to tell us what that means?
Well, that is really in the context.
That's just a way for us to remember this.
Okay. Yeah. Okay. So, well, I think they'll remember.
Yeah.
But, cells that fire together, wire together is, a saying that really reflects the idea that if connections are used all together at the same time, sort of very robustly, they will strengthen. And if they're not used at a time when other connections are being used, then they will weaken. And that, you just think about the two eyes' inputs. Fire, wire. So you want to make neurons that are getting input from both eyes. So you want both eyes to be working together to really build those connections in the visual cortex. So that's a fire-wire situation. So if one eye is weaker than the other, it's not going to be used. When the strong eye is being used, that's out of sync, lose your link. So that's the kind of concept there. And now we know that those fire-wire means strengthening, and out of sync, lose your link means pruning. And we know a lot more about that at the cellular level.
Carl, I have a lot of questions from the audience already. Some of them are, as you probably are seeing as well, I'm gathering some popular support here, and I'm going to come to some of these in just a minute, but a couple of things. Let's make the connection then to the question that all of us in this room want to know, because we all have brains, and we all worry about our brains. We all worry about what our brains will be 5, 10 years from now. We worry about, has anybody in the room tried for a name and it doesn't just quite come the way it used to? You too? Oh, please say it ain't so. So with these discoveries that you have made, Carla, are we closer to figuring out why our memories fail us? And that pill that you're talking about, is that anywhere on the horizon? Or are we still 10, 20, 50 years away from that?
Well, I mean, we're definitely closer than we were, 10 years ago. There's no question about that. What I worry about is that I think our colleagues who have been conducting the clinical trials for Alzheimer's may be intervening too late.
What do you mean?
And what I mean by that is, well, let's just think about Alzheimer's for a minute. And let's think about the plasticity of our brains also for a minute, because when, I can talk about my own, family, but when a patient comes to the clinic with cognitive loss and then is scanned, let's say.
The brain is scanned.
The brain is scanned, and the doctor tries to work on what is the reason for the cognitive loss. And these beta amyloid plaques are discovered. My question is it already too late to intervene and treat. Now, why do I ask that question? Because, as I said, your memories are stored at synapses. And also, our brains continue to learn and are plastic, even as adults. And plasticity means new connections are formed and others are removed. That's what brain plasticity is. When you do one of those brain games and you learn something new, that's brain plasticity, and you're adding synapses and or removing synapses.
With the brain game.
With the brain, well, we could talk about that later too, but with learning.
With learning.
And training. So at the point, so your brain is constantly able to remodel with pruning and strengthening of synapses. And at the point when we show up in a clinic with memory loss, the question is, how many synapses have we already lost? How many have already been pruned? And it's, I think, a really profound question, and I don't think anyone has the answer to that question yet, because we really can't count in the patient those synapses, because our methods of looking for synapses are not sensitive enough yet to study the synapses in the living brain. But we can check later in autopsy material, so people are very generous and have given their brains to science so that synapses can be counted just like they were counted here in the developing brain. And already at that time, it's seen that there are a lot of synapses lost. And later, at later times, nerve cells are lost. So the question is, if we could intervene earlier, maybe we could actually save the synapses by preventing pruning. But we would have to know if there is excessive pruning in Alzheimer's, and we would have to understand what the mechanism of that pruning is.
So we have more understanding to do, clearly, before
Lots
we can
more.
do that. When you talk about intervening earlier, would this be, you talk about the pill, right? Would this be something that we're doing in adolescence, that we're doing as we turn 40, that we're doing when we first detect some change? When might that intervention happen?
Right. Well, certainly, so let's just go to the trials, the current trials. There are still several trials that are in progress, so there's still hope. The idea of the current trials is to, at the time when patients appear with serious cognitive loss, there are plaques that have formed. And the idea is that these plaques are very damaging to the nerve cells and cause the nerve cells to die, which they definitely do. And so the idea is to bust up the plaques with some of these drugs, and these drugs are apparently quite good at busting up the plaques, and yet patients don't seem to really respond the way we all hope that they would. And I say I am not a clinician, I did not design any drugs, but I'm really hoping, like all of you, that these drugs are going to work. So we have to ask, why is it that even though the drugs bust up the plaques, the patients don't get a lot better? There's some improvement, especially with some of the newer trials, but it's really disappointing. So an answer is that it's too late because many of these synapses have already been lost. So now how early should we go? And I think we don't really know. We need to know when, even before these plaques form, when does the disease start? When does Alzheimer's disease start? So right now there's several clinical trials that have decided to go even earlier, studying families that tragically have genes that are known to cause early onset Alzheimer's. So there's several important trials that have started earlier, and I think this is really good. But is it early enough? Because if you have a gene that produces material that will eventually cause the plaques, this is called beta amyloid, then that material may be produced very, very early in life. So how early should we go? So my answer is definitely earlier than the current trials. There's no question about that.
Carla, you've spoken a lot about Alzheimer's. I have a 94-year-old mother-in-law. She's amazing. Our grandson, my nephew, is going to get married next month. And she almost decided not to go. because she's horrified at how difficult it is for her to recall the names of even the people she's been with all her life. She's not disoriented. She doesn't fit, and her diagnosis is mild or moderate cognitive loss. All right? So, but is that the same thing that's happening in the kind of forgetfulness that most of us experience, and now that we're living so much longer, we're going to experience more?
Right.
And does this maybe hold some answers to that?
Well, I think it does. I think certainly it holds some answers to that. I mean, there's really serious, cognitive loss, and there's mild cognitive loss. And, the curve that I put up in the beginning suggests that we're all, as a process of aging, we will all suffer some cognitive loss. So I absolutely hope that she is somebody who will have a very healthy brain.
But this is happening also because of this her synaptic pruning.
That's a hypothesis only in the sense that, we can't look in her brain until after she's gone.
Yeah, I asked you.
And so we don't want to look before.
She's terrific, but she wouldn't go quite that far.
No, exactly. But I think it really poses a huge challenge that should be met currently, and I think, technology-wise, we could do it with some very inspired science and engineering
Carla,
that would let us look at the synapses in her brain. And
question from the room here, and it's got the most votes while we're talking about this. What role can music therapy play in the development of new synapses? So can new synapses be developed? You're looking for the molecule, right?
Right.
But how about music therapy?
Well, I think, like I said earlier, I think using your brains, and if you don't fall asleep at this talk and this event, you will have used your synapses in a really good way. You will have added, if you remember, you will have added quite a lot of synapses, and we know that those synapses can store memories from studies in the animal models, where if you then remove the learned synapses, the mice forget. So you can learn new things, you add more synapses, and that is really the process.
So while we're on that, as a related question.
But maybe, the music question is a really important question in the sense that pretty much in a healthy brain, pretty much any exercise that you do to add learning is an exercise worth doing. But sitting and playing a game over and over again, unless it's really cognitively exciting and challenging, is not necessarily going to be helpful.
And you just actually mentioned the word exercise. This is in the other question. Is it true that exercise, meaning physical exercise, helps promote brain activity that can counteract dementia?
Well, physical exercise, right now, most clinicians say that the best thing you can do for your brain, other than keep your brain going by challenging and thinking and going to lectures and, doing things and not being a couch potato and passively watching TV, the best thing you can do really is to keep yourself in good cardiovascular health. And there are a number of reasons why that's very important. The first is just that your brain requires a huge amount of oxygen to function normally, and so you want to really have good cardiovascular health for that reason. The second reason is kind of more interesting, and that is that it's thought that inflammation may contribute, it's known that inflammation contributes to cardiovascular disease, and it's now thought that inflammation may also contribute to cognitive disorders and cognitive loss. And people don't really understand the relationship between inflammation in your body and what might drive neurodegenerative disorders in your brain, but it's quite clear that people who really are healthy aging are also in very good cardiovascular conditions. So that is why it's probably extremely important. It's a very good question about how the immune system and inflammation really intersect with brain function, and it's very exciting. We don't know anything about it right now. We don't know a lot.
Aside from being this astonishing scientist and winning the Kavli Prize, what do you do for exercise to keep your brain fresh?
I'm really bad right now. But when I have time, actually, I'm very good at avoiding exercise and in making up excuses. I'm so good that I figured out how every time I pass my my health club on the way home from work, I have an excuse not to stop. So actually, I work with a personal trainer twice a week,
Oh, wow.
and I do yoga on
Well,
the
that's
other
good.
days.
That-
So
that
yeah, but
works.
when I travel, I don't do that. So.
Another question, how much do genetics like APOE4 play into the susceptibility of Alzheimer's prevalence? And what sort of prevention measures can be taken? That has a lot of votes.
That's a really good question. Well, so ApoE, this particular version of apolipoprotein, which is very important actually, again, for your cardiovascular health. This particular version, for a reason really that I think people don't truly understand, seems to create a higher risk of developing dementia. It's not just ApoE, so there are a number of gene variants that have been discovered that put you at higher risk for dementia. And that's an extremely bad scenario. So another one is a mutation, a change in this APP protein that actually also gives rise to extra beta amyloid, which causes accumulation in the plaques. And in families with ApoE4 version or with APP, versions of APP that seem to allow for the plaques to build up very quickly, there's a high risk of early onset dementia. And in fact, these families have become the target for the administration of earlier drugs by pharma. So these families, while having this higher risk, are also in a major way contributing to our understanding and maybe even the solution and treatment of Alzheimer's disease. So it's on the one hand bad news, and on the one hand really we have to thank all of them for what their genetics are telling us and what might allow us to all have a pill someday.
Carla, every scientist who does original, difficult work encounters significant challenges, obstacles along the way. What are some of the biggest challenges that you've encountered in this work?
Well, I think for me, one of the biggest challenges is that we keep finding weird stuff in my lab, and then it takes about 10 or 20 years for people to see that, oh yeah, that's actually pretty good, and that's right, and other people go and they will reproduce the work. For example, the early signaling from the eye to the brain, and the finding that the brain actually needs that signaling in order for the adult pattern of connections to emerge was really, people were very skeptical about it. How could the embryo be sending these signals to the brain? It just seems too dangerous, and you should really hardwire everything in order to make sure it all develops properly. But then, as I said, if we hardwire everything, the brain can't learn to use both eyes together, for example. That has to happen through use. So I think for me, those were very hard periods. And I feel very lucky that I have many mentors, and my parents taught me, they gave me some resilience somehow. I don't know how that happened, but so instead of just being sad and depressed, I got mad. And so I think that's very helpful.
What did you get mad at?
Well, I got mad at my field.
You got mad at your field?
Yeah,
What did you get
my
mad
colleagues.
at?
Well, mainly just for them. I think when I started as a scientist, I really thought that the whole idea was you're supposed to make new discoveries, and then I figured, okay, everybody's just going to say, oh, that's really cool, nice going, But science is a very deliberate process, and it has to be replicated. And for people to even replicate the work, they actually have to believe enough in it to spend time showing, that it's correct, and then taking off from it. So I think that surprised me. I mean, I wasn't very sophisticated. I was very idealistic.
There's a question that comes right out of that from a lot of people. As a prominent woman in science, do you feel that you had to be your own advocate to get here? Did you have to overcome adversity from male colleagues for your work to get the recognition it deserves?
Well, I think I had to be strong. And what I mean by that is, I I did not feel as though I was, that there was adversity from my male colleagues. In fact, when I was an undergraduate and a graduate student with my mentors, both of whom were men, David Hubel and Torsten Wiesel, I felt enormous support from them. And I actually think they gave me some of my sort of self-reliance and self-confidence.
Do you have any women role models when you were?
Well, no, and I mean, I think that's the other thing. I mean, there were no women role models. There were like 2 women. You know, we went to Stanford, there were like 2 women. We knew each other, we got to be really good friends. I mean, I see Maxine Singer, she was like, there was 1 woman.
You do a lot of mentoring now of young women
Yes,
scientists.
but what I say now is, I mean, so we had our mentors, I think for me, many of my mentors and people who really supported my career were fabulous men. I wish I had had some womentors because, you know, I mean, I really didn't know when to have kids, and I waited too long, and I couldn't have them. I was too old. You know, I thought I could just do it any time. And I think if there'd been a few women there as, you know, pioneers, I would have, I think I would have had them earlier. That would have been straightforward. But anyhow, now I think it's a different situation because there are many women. There should be more, by the way, but there are many. And what's so fantastic right now is that the generation or 2 younger than me feel really great about starting their careers as assistant professors and also starting their families at a good time. And it's really great. But what I tell students now is they still need, both men and women, still need to build a framework and a network both among their friends and also, very important, among senior mentors. And because that framework can really support them when they encounter adversity.
Sure.
So, you know, the problem isn't solved now, and a lot of work still has to happen. But I think that kind of framework is something that is now available that was not available
Carla,
before.
another question from the audience. Is there a link between traumatic events and the onset of Alzheimer's?
So I don't know the answer to that question. I don't know if, but I can say that, again, it is actually known that stress can alter your immune system's ability to regulate inflammation. And there could be that kind of connection, but it's not something that I know that has been really formally studied. But maybe someone in the audience will write back to you and say that they know.
How about this? Do the brain development processes that your lab studies that you've talked about here continue throughout childhood is the question? Or are the brain connections that we build later in life reliant on different physiological processes?
Oh, that's a great question. So again, so if we think about the mechanisms from animal models where those connections and mechanisms can be studied in detail, there are 2 things that happen with ongoing development. The first is you can kind of think of this pruning process as controlled by a kind of rheostat, and that can be dialed down. So it goes on throughout life, and there's evidence that the mechanisms in the adult are the same, but there's some kind of a braking system that's been put on top of those pruning mechanisms. And so that then makes the quality of learning different later. The other thing is that new learning mechanisms, new molecules are turned on and appear in the adult brain. And so they're both new kinds of brakes and new kinds of, you could call them, I guess, accelerators.
Right.
So there is a fundamental theme that's running throughout life, but it's regulated. I don't know if that answers the question. So it's not like the adult brain is completely different from the child's brain. No, it's a continuation with certain regulators put on top of it.
Where's your field going? Where is this going?
Well, I think it's going in several directions. First, I want the pill, of course. So, I mean, I think the field, first of all, how can we relate the changes and these mechanisms that we study at the molecular level and in mice models, how can we actually relate them to global circuit changes that underlie behavior? Huge question. And how can we study these mechanisms and molecules in our own brains, in human brains? So I think the field is going in those directions. So it's very exciting. So there are new methods really for studying how changes in learning can result in changes in circuits, and you can actually see the changes in the circuits using methods that allow you to look at hundreds or thousands of nerve cells and their connections at the same time. These are being developed now. Part of it is thanks to the BRAIN initiative that was initiated under President Obama and is now being carried out under the NIH. So there's actually funding for trying to develop many of these new methods, which is very exciting. The other is to really ask, how can we study these mechanisms and molecules in the human brain? And obviously, we either would be great to develop new imaging methods. The strength of the human genome analysis allows us to relate the molecules we discover in the mouse to similar molecules in the human brain that in principle could be studied. So the molecules we study in the mouse, we can find them in the human, which is really exciting, but now what do we do? Yeah. So, right? So the most amazing thing now is that people can grow little organoids of human neurons. They're called like mini brains. They can take your skin cells, coax them to become neurons, put the neurons together, and miraculously they recognize each other and can assemble to form little teeny organoids, but they're human neurons. They're just little teeny things floating around in a dish. So we can study the molecules and mechanisms because the neurons hook up with each other. So it's really quite amazing. So this offers huge hope for therapy. And in fact, some of the first mini brains that were made from the skin cells of a child with Timothy syndrome, a severe form of autism. And what was discovered in the little mini brain was a possible cause for the autism in the child because there was a problem with the nerve cells assembling in the right place and also in communicating with each other through calcium signaling. So it was kind of a proof of principle. And you could imagine then actually trying out drugs on the mini brains. So maybe you wouldn't have to try out the drugs on humans first. And the mice might not be good models for humans, as we've learned recently. So I see the fields going in really exciting, crazy ways that have been driven by the investment in really fundamental science that's been made in the past.
Investment in fundamental science, but you have said that you're not sure that your research would have been funded today.
Yeah, that's true. It certainly wouldn't have been funded today. I think I would have had to, first of all, I mean, my first grant that I got from the NIH, I had no preliminary results at all. So they took a big risk. So I really, really plead with everyone to think about allowing great, crazy ideas to be tried out because you never know where they'll go. But what I think is that, I was funded to study the eye-to-brain connection during development. And people were patient with me. The NIH was patient with me. And they continued to fund me. And then I was surprised to find myself thinking about neurodegeneration and a real clinical application, a potential clinical application. So you just don't know where the basic, curiosity-driven research is going to lead. And I'm very grateful that I've had the chance to do this.
So before we go, we're almost out of time here, but Carla, you've been doing this for a few decades now. How did you get into science? What got you so excited? How did you get into this?
Yeah, sometimes I sort of say what kept me from being dissuaded from getting into science. You know what I mean? It's the other side of the coin because I mean, I think what happened is I think children are fundamentally excited about science, and they're fundamentally excited about the scientific method. And they're always asking, why, right? I mean, they're always asking these questions. And so I think for girls especially, often there's so much social pressure. And I think for me, I just was not diverted from asking the scientific questions and being rewarded. So my dad was an aeronautical engineer, and he contributed his team contributed some of the first engineering for the lunar modules that, for the space race. This was really important. And I was a little girl then. My mom was an artist, probably says something about the union of my interest in the visual system and science. But they encouraged me. They didn't discourage me, and that was really important.
Well, let you leave us with your thought for your next big breakthrough. What are you hoping that
Okay,
your
well,
next Kavli Prize will be all
my
about?
My next Kavli Prize, okay. Oh, this was not planned, but I have the answer. Wait, I mean, it's a silly answer, but it's serious, by the way.
Silly but serious.
Yeah, silly but serious. Okay, here it is. Here's the answer. So that's my grandmother, and that's me when I was about 4 years old. And I think, we would love to see some of our fundamental work really reduced to practice and used therapeutically. And if it's not ours, then the plea would be to really listen to some of the newer ideas that have come out of the study of fundamental brain development and understanding how synapses are formed, maintained, and broken, and use those ideas at earlier times to treat neurological disorders. And this doesn't just have to be of aging, but it could also be of learning and memory in young children with autism or other disorders.
Well, Carla, let me thank you for all your incredible work, for contributing to our Synapses here this evening. Bravo. If there is one thing that gives us hope in a crazy world, it is the progress that scientists make to discover, to find new knowledge, and to move humanity forward. So thanks to Kavli, thanks to Carnegie, thanks to you, and Eric, back over to you.
Well, first of all, I want to thank both of you for a terrific pairing and some great conversation. Thank everybody for coming. Pay attention to Carnegie Science. We have these types of lectures on a regular basis, and we'd love to have you come back. So thank you all for coming, and good night.
Thank you.